Hello, everyone, and thank you for joining me today. Over the past few years, there have been much activity around implementing the quality by design principles for analytical method development. At Waters, we have also been applying this approach for developing methods using various software platforms. In this presentation, we will give an example of method development application that employs the quality by design principles using statistical software for data analysis and mathematical modeling. In this presentation, the focus will be on developing a method for budesonide, formoterol, and related substances. The cost of poor quality for pharma is very significant, and this can cause a lot of losses, including recalls and shortages of medications, kappa overload, and loss of revenue. Analytical methods play a very critical role in the quality process and the quality of medications. So it is critical that analytical methods are built in a very high quality way to make robust methods reproducible, fit for purpose, and very precise methods. While there are many ways to develop analytical methods, the focus of this presentation on implementing the analytical quality by design for developing methods. The analytical quality by design is a systematic approach to method development that starts with the predefined objectives. In this approach, we define a region where chromatographic space, where small a region of chromatographic space where small changes will not affect the results. This region is known as method operable design region. We define this space by performing design of experiments. We evaluate and sample the space efficiently. As a result, we gain knowledge about method performance characteristics and study factor limitations. A quality by design approach uses statistical software to generate experimental designs and to study interactions of different factors. This approach is highly comprehensive. It can be automated and offers the most knowledge about a method and its robustness. However, it requires more processing and confirmation of final results. For this application, we will be discussing, as stated earlier, Symbicort, the analysis of Symbicort components. Symbicort is a medication that is available by prescription as an inhaler. It consists of two active pharmaceutical ingredients, budesonide and formoterol. These two components treat different symptoms of asthma. So in order to comply with regulatory agencies, it's important to have a method for the analysis of this drug medication. The current methods for the analysis of budesonide. There is a USP method for the analysis of budesonide, and this method uses reverse phase HPLC method. It uses a 3 micrometer C18 column, that is 250 millimeter. The total run time for this method is 70 minutes, which makes it very time-consuming and very solvent-intensive method. Additionally, there is currently no USP method that describes the analysis of budesonide and formoterol in a single pharmaceutical formulation. So the goal of this work was to develop a method that is MS compatible for the analysis of budesonide, formoterol, and related substances using Fusion QBD as software for the analysis. Another important challenge with this application is the fact that budesonide exists as a pair of epimers, and resolving these two peaks from each other is normally challenging. So we thought this would be an interesting application to see if using tools would help us to make this separation easier. All right. So with method development approach, if the analytical quality by design focuses on various steps to follow when you do method development. The first step in analytical quality by design is setting method goals, where you start by defining the method goals for the final method. The next step is the risk assessment, and in risk assessment, we will talk we will talk more details about each one of these steps in the next slide. Risk assessment, you identify critical method parameters that could significantly affect the performance of the goals. Screening, where we will be doing we will try to screen the effect of major chromatographic effectors on chromatography. And optimization is where we do some sort of fine-tuning of the final method to make sure that we build a robust method that can be fit for purpose and reproducible and accurate. So if we go to the method goals, the method goals are the analytical objectives that describe the intended purpose of the method, what will be measured, and the performance criteria for the measurements. For the budesonide, formoterol, and related substances, the method goals included performance characteristics, such as USP resolution of at least 2 between all the analyzed, USP tailing of less than 1.4 for all the analyzed, percentage RSD of peak area of less than 2 for all peaks, and percentage of retention time of less than 1 for all peaks. So these are the goals that we set for this method. The next step, according to, or not according, that goes in compliance with the AQBD requirements, is to do risk assessment. And in risk assessment, critical method parameters are identified and assessed for the highest impact on the quality of the data generated. The high-risk parameters that may affect the method's ability to meet the goals are assessed based on the sound chromatographic science, literature, review, previous knowledge, and experience. Risk assessment is often done by using fishbone diagram to visually show which method parameters have the most impact on the method performance. For budesonide, formoterol, and related substances, the critical method parameters are indicated in yellow. The parameters include column chemistry, mobile phase, buffer, and solvent type were identified to have the greatest impact on the performance. Therefore, they were studied in the screening phase of the method development. Other chromatographic parameters, including flow rate, gradient time, and slope, injection volume, can affect the resolution between analytes. These parameters can easily be controlled. Therefore, they were not considered critical. Design of experiment for screening and optimization studies were performed using Fusion QBD, which is a software that is commonly used for statistical design of experiment method development. Separate DOE studies were created for screening and optimization phases. DOE at each phase was exported to the Empower software for data acquisition. After completing the runs, we processed data in Empower and imported results to Fusion QBD for statistical analysis and modeling. The numerical search for best conditions was also performed. As for system, we used an H-class system that is coupled with PDA and QDA for mass detection. And we also use a column manager that allows us to use multi-columns for screening. This slide shows the workflow within Fusion software, and we will go over these steps in more details in the next slides. But in general, the Fusion QBD method development workflow starts with some sort of preliminary experiments, and the reason for these experiments is to, if you don't have a primary method that you can use for further screening, you can start from scratch and do some preliminary experiments to find the optimum wavelength, to find the conditions where all of the compounds are eluted within a reasonable time frame. The next step within Fusion software is screening, and at this step, what we do is typically try to screen the most important chromatographic variables, the highest risk factors for the performance of the method, such as the most important one is the chemistry of columns at this stage and mobile phases. pH also is a major influencer in the performance of chromatography. The next step is optimization. And optimization, typically what is normally done is fine-tuning of the less important chromatographic factors to achieve the best separation between the compounds and the most robust method. And finally, characterizing the final design space and creating a method operable design region, as we stated earlier, where small changes to conditions do not significantly change the performance of the method. So in Fusion, the way it treats chromatographic factors, as either numerical factors or categorical factors. So numerical factors are factors with levels which are represented by numbers on a continuous scale, which has meaning, and for which intermediate levels between these two numbers also has a meaning. For example, if you have a temperature of 30 and temperature of 40, a temperature of 35 would have a meaning. That is a numerical factor. On the other hand, there are some factors that are categorical, where where they are discontinuous and intermediate values are not particularly meaningful. For example, the stationary phase chemistry. If you have a phenyl column and a C18 column, there is nothing in between the two columns. A major advantage for Fusion QBD is that it minimizes the number of experiments that are needed to obtain a knowledge about, a very comprehensive knowledge about the method. So this is an example where if a full factorial design of experiments needed to be done for 5 temperatures at 5 different gradient times, we will need 25 methods. Using Fusion and doing statistical sampling, this number can be lowered to 13, which is significant reduction in number of experiments, almost 50% reduction in number of experiments. So Fusion does statistical sampling to reduce the number of experiments that are required to create a design space. Now, if a third variable is added to this study, such as the pH, Fusion would still apply the statistical sampling to create the design with minimum number of experiments required. However, if a fourth variable that was added, and this variable is categorical, the same number of experiments will be repeated across the different categorical variables. With this, Fusion would still significantly save on time by reducing the number of experiments that would normally be needed if a full factorial design is implemented. So for preliminary experiments, what we did, we chose a couple columns, HSST3 and the BHC18, and the basic goal of this experiment is to find appropriate wavelength that all analytes would have decent absorption and much interference from the mobile phase, and also to make sure that all peaks will elute before the end of the run. So it's a very limited number of experiments, and the purpose of this experiment just, as stated earlier, to find initially conditions that can be used for further screening experiments. So this is a chromatogram for initial chemistry screening experiment. Note that in this chromatogram that there are several peaks are not optimal. And again, the purpose of this experiment is just to have starting point conditions, and the peaks are not expected to be perfect at this stage of the study. Based on the preliminary screening experiment, we go next to the most important step of the study, which is the screening. And in the screening experiment, the goal is to screen the most important factors that could affect the separations. So in this experiment, as we are using an H-class system, which is a high-performance instrument, low dispersion volume, we use sub-2 micron columns, 2.1 millimeter columns, and they were all 5 centimeters, so we can achieve fast separations. And the gradient times were between 5 to 15 minutes. Online mixing from the acid and base was used at this stage of the study to achieve the desired pHs, although this is not the optimum way of preparing buffers, but at this stage of the study, which is only to screen the best conditions, it is accepted to use this kind of online mixing for the desired pH. Another important thing is that the gradient slope in fusion can be achieved by two ways, either by changing the organic solvent composition or the gradient time. Fusion allows you to do only one at a time because it wouldn't be able to handle the small differences between the two approaches. Wide ranges of chemistries, which is the most important factor in a screening experiment, was explored in this study, and we'll talk more details about these chemistries in the next slide. So these are the 5 columns that we have used in this study, and as we said earlier, that for method development, the goal is to choose columns that provide the widest range of selectivity to increase the chances of success. So we used 5 columns for this. The first column we used was a BHC18, which is one of the most popular LC columns, and it's available and does have a wide range of pH range. The second column was a shield reverse phase C18. It does offer alternative selectivity to C18 as it contains embedded polar groups that can change retentivity. The third column was a cortex T3 column, which is a superficially porous column that gives increased retention for polar analyze. The fourth column was another superficially porous column, which is the cortex phenyl, but it does offer unique selectivity with phenyl groups and pi interactions. The last column was the PFP column, and it does have completely different selectivity, and it was interesting to try this column too, as we said earlier, to have the widest range of selectivities. It's important to note here that it's important to choose the right geometry of columns when depending on the instrument you are using, and since we used an Acuity H-class system, the appropriate geometry is 2.1 millimeter columns, 1.x micrometer particle. So for the screening experiment, this slide shows the various conditions that were studied in this screening experiment. We used categorical factors, which are the 5 columns, and we also have used the numerical variables, which are the different pHs of 2, 2.5, 2.9, 3.5, and 4.1. The flow rate was constant at 0.5 ml per minute, and the injection volume was 3 microliters. And the open temperature was also constant at 40 degrees Celsius. The goal here was to keep the number of experiments manageable, so you get the maximum, the most reliable results out of fusion when you cap the number of experiments to 40 or 50 experiments. An important feature in fusion method development software is that it's highly compatible with a wide range of instruments, Waters and other vendor instruments, and it is also compatible with various modules and various detection setups. It also automates the whole process of exporting and creating all the methods within Empower. It also creates all the required conditioning and equilibration steps that are needed for the experiment. This feature is very useful, not only because it saves time from manually entering and making all of these methods, but also it eliminates any transcription error. It's important to note here that although Fusion interacts seamlessly with Empower, it wouldn't log into Empower without asking you for your login credentials. It won't automatically log into Empower. So it goes with the level of permission you have for Empower to log in. This slide is a chromatogram from the screening experiment. It showed the best looking chromatogram on each one of these columns from the screening experiment. And when the experiments are done, you have to do the processing in Empower. And this is done normally by browsing through the chromatograms, finding the best looking chromatogram, and then creating a processing method based on that chromatogram, then apply this method to the other chromatograms. Based on the results, as can be seen from the best looking chromatogram on the top chromatogram, it was clear that there are major issues of these separations that need to be addressed. The first issue we had is that the formoterol peak is very broad peak, and the USP tailing is really high. So this needs to be addressed if we want to get a high quality method at the end. The other important issue that a cluster of peaks at the end are not very well resolved. So the end of these results showed us that there is a promise, but there are some issues need to be addressed to get a final good method. It's also important to note here that using a mass detection, the QDA detector, was very useful in this exercise as it allowed me to know which peak is which at the different chromatographic conditions. It would be much harder if you don't have mass detector to know which peak is which, considering the different selectivities and different elution orders when different chromatographic conditions are utilized. So the first issue I wanted to address is the formoterol peak shape. And to address this issue, we made an adjustment to the design of experiment. This adjustment was using high pH aqueous mobile phase buffers. And the reason for this adjustment is the fact that formoterol is a base, and it will be logical that high pH mobile phases could help improving the peak shape of this analyte. We only used one column for this screening, which is the best column we obtained from the previous screening experiment, the BHC18 column. It also, this column was also used because it is very compatible with a wide range of pHs, including the high pH that we are about to use in this experiment. Another important note here that the calibration curves for the online pH mixing that were done for fusion software were all performed on Acuity H class. And as we are using Acuity H class, it means that these predicted pHs and the mixing would be very accurate. So, as we said in the previous slide, this was done using the BHCATN column. And in this experiment, we screened a wide range of pHs ranging from 6.7 to 10.7 to see if improvements can be achieved for the peak shape of formoterol. All other results were maintained the same as in the previous experiment, including oven temperature, flow rate, and the gradient profile. So the results of this experiment showed that formoterol peak shape has significantly improved when high pH mobile phases were used. For example, the tailing factor, as can be seen in this chromatogram, was less than 1.3 for this chromatogram, and for 10 other chromatograms out of the 15 experiments that were performed for the screening experiment. The figure on the left of the screen image is a screen image from Fusion, and it shows a numerical search for the best overall answer. And the white area represents the combination of experimental conditions where the performance goals are met, and the shaded area represents the combination of experimental conditions where the performance goals were not met. It can be seen here that there is a wide range of experimental conditions that where baseline resolution can be achieved for all analytes. So the results of this screening experiment was successful in addressing the peak tailing of formoterol and giving some promise for good separations of the cluster of the peaks. While this method provided good separations, further optimization to reduce, hopefully to reduce the runtime, but most importantly to get better peak shapes and better resolutions would be important to pursue. So the next step was optimization. And the goal of optimization is to identify a method with optimum performance and robustness. In this optimization, we used the BHCATN column, and we used various pH buffers. We varied the less important chromatographic parameters such as flow rate and oven temperature. And we used the 3 different pHs. And the pHs that were used in this step were all pre-mixed buffers for more accurate results. And these buffers were employed using the switch valve on the H-class system that allows for using different solvents at the D valve. And the variables that we used for this experiment were the gradient time, 15 to 20 minutes, and the flow rate, 0.3 to 5 minutes, and the column temperature. It's important to note here that we used a 10 centimeter column for the optimization experiment, given that the 5 centimeter column did not give us the desired separation between challenging peaks such as the epimer peaks. We thought that using a longer column would give us higher efficiency, and this would increase the chances of success, achieving the performance goals. And just like in screening, we perform the numerical search for best method, then we explore the acceptable performance region for that method. Here we can visually examine the area of robustness over the range of method conditions. If we look at the graph on the right side of the slide, the x and y-axis represent method numeric variables. In this case, the x-axis represents the oven temperature, and the y-axis represents the gradient time. The box or rectangle within the graph is referred to as the design space or method operable design region, which represents a region of an acceptable and robust method performance. The target T at the center point is the final method, and the prediction points A through D around that box represent the confidence limits at which method meets the performance goals for USB resolution, peak tailing, etc. You can export the prediction points to Empower software for verification runs of your experimental conditions. Overall, again, the unshaded area indicates the conditions under which the method meets the performance goals, while the shaded area are the conditions where the performance goals are not met. And it's important to note here that this is interactive display, so where you put the precursor on the figure in Fusion, it shows you interactive display of the predicted chromatogram of these conditions. Another important feature I noticed personally when using Fusion that the predictions of Fusion are pretty accurate, and this is an example of this table is an example of critical pair resolution at various experimental conditions, and the predicted results has always been very close to the actual results. And I've done over 25 verification runs, and the predicted results have always been within less than 2% of the actual results for all of these 25 verification runs. So it's normally pretty accurate. So as I said earlier, you can export the final method to Empower and do some sort of verification runs for this method. And this slide shows multiple injections of a final method showing that the method is robust and the system suitability criteria are met. The method performance goals that we set at the beginning are all met. So USB tailing was always less than 1.2 at the final method. USB resolution was always over 2.4 for all analyzed, although the goal was to have it over 2. Percentage RD of peak area have always been for all analyzed less than 1%, and the percentage RZ for retention times have always been less than 1% for all analyzed. So the final method showed to be very robust and under a very wide range of chromatographic conditions. The resolution maps in Fusion enable a prediction of the resolution between the least resolved peaks. The colors represent the resolution value between peaks. If you look at the legend, the red color indicates higher resolution. An important thing to note on this slide is that the pH and the temperature have very important effect on the separation. So For example, at a temperature of 32 degrees here, you can get a baseline separation, you can get a resolution of over 2 for all analyzed. But if you go to a temperature of 48, you lose this resolution and you hardly get, you don't get any baseline resolution for the critical pair. So with these resolution maps, they are very informative and they would give you information that you wouldn't be able to get without this visual representation of these data. So in summary, I hope that this presentation was able to show you that efficient method developments can be achieved using water systems and fusion software. It allows for risk assessment. It allows for defining method operable design region. It also identifies the variables and the critical variables that could affect the separation of chromatography. It also shows that fusion can be very useful for method development as it automates the method development process by creating all the desired methods for all analyze, for all experiments. Another important thing is that this method, this work, allows us to separate analytes that have never been separated in literature with this success, including the challenging pairs of epimers for budesonide and budesonide-related compounds. Another important thing to note is that the QBD development software allows for rapid separation for all analytes because it uses statistical sampling to reduce the number of experiments needed to get a comprehensive knowledge about the various chromatographic variables and their effect on chromatography. And with that, I would like to thank you all for joining me today.